Hi there. In this video, I'll introduce genomes and talk about genomes of RNA viruses and DNA viruses. And we'll be sort of progressing through genome evolution, starting with these fairly simple genomes. And then in a subsequent video, I'll talk about bacterial and archaeal genomes, and then parasites, endosymbionts, and organelles in an additional video, and then the final video will be on genomes of eukaryotes. In some of the previous videos, I've discussed different types of genes, and pretty much on their own rather than in combination. So in this set of videos, I'll talk about how genes combine and interact within genomes. Although I'm going to focus primarily on genomes, that is the complete DNA sequence of an organism, we also will discuss a little bit about the proteome, that is the proteins that are encoded in those genes and RNA molecules, and talk a little bit about the transcriptome, which is the complete set of RNA sequences of all the transcripts. And I won't talk quite as much about the metabolome, which takes some different types of techniques, and that's the complete list of metabolic pathways and enzymes of an organism. There's also an RNA ome and other types of omes that have been defined in the past decade or so. So when we talk about genomes, you can define it in several different ways. So the total genome is the chromosomal DNA, or RNA for RNA viruses, plus the mitochondrial DNA if the organism has mitochondria, plus chloroplast DNA if it has chloroplasts, and other types of DNA also. For eukaryotes and anything that has an organelle that has DNA in it, you can also just refer to the nuclear genome of everything in the nucleus, a separate mitochondrial genome, a plastid genome, or other organelle genome as well. You can talk about those separately. So here are a couple of examples. Bacterial genome or archaeal would consist of the chromosome, and bacteria can have 1 to 5 and possibly more chromosomes, so all of that together. If there are extra chromosomal elements, also called ECEs or plasmids, those can also be discussed separately or together with the genome. Some bacteria have multiple chromosomes, as I said. Some have small elements that are extra chromosomal, but have quite a few genes on them. So those are also present in many bacteria. The DNA viruses usually have from 1 to a few DNA chromosomes. They can be single-stranded or double-stranded. RNA viruses can have from 1 to, I think influenza has 8 separate RNA chromosomes, single-stranded. There are some double-stranded RNA viruses as well, and those are complete genomes for those. And then you get to the eukaryotes, where there's always a nuclear genome consisting of 1 to sometimes thousands of chromosomes. There are mitochondrial genomes often, but not always, so that can be discussed separately. Plastid genomes for plants. And then other types of genomes, other types of organelles in protists, where you have a red alga or a green alga inside of another eukaryotic cell. And then those would have their own genomes consisting of sometimes nucleomorphs with DNA in them. Mainly not mitochondrial, but you can find the mitochondrial DNA in the nucleus. And often, almost always, a chloroplast genome, some of which or all of which can be found in the nucleus. So several types of ways to talk about the genome, which is essentially the total DNA, in the case of most organisms, of a cell, or RNA in the case of an RNA virus. Here's a graph of various genome sizes for groups of organisms, and this is a log scale down here. So here's 1,000 nucleotides, here's 1,000,000 nucleotides, there's 1,000,000,000 nucleotides, etc. Starting with RNA viruses, which are the smallest among the organisms, and then DNA viruses, plastids or chloroplasts, mitochondria, and then bacteria, archaea, and then all of these up here are eukaryotes. Starting with the protists, and that's a very broad scale, primarily because it encompasses a large number of very diverse phyla or organisms. And then plants here, algae, mosses, lycophytes, monilophytes, gymnosperms, and angiosperms, fungi, and what are called the opisthocons, which are fungi, and then animals up here, invertebrates, fishes, amphibians, reptiles, dinosaurs, birds, and mammals. And you might be surprised that I put dinosaurs up there. How that's done is that the nucleus size relates directly to the amount of DNA in the nucleus. And in some dinosaur fossils, you can actually see cellular details, including nuclei. So you measure those, and you can get the approximate sizes of the genome of those organisms. And when that's been done, they fit right where they should, sort of in between reptiles and birds. Birds are one of the last living lineages off of that dinosaur line, and then reptiles led to the formation of, or the evolution of, dinosaurs too. So those are accurately placed there. Also on this table, you notice that some of these are white, and those are obligate parasites, meaning RNAs need host cells. RNA viruses, DNA viruses need host cells, so they're parasitic. Also, there are many bacteria and archaea that are parasitic as well, so those are down here. You'll note that the parasitic ones have the smaller genomes of each one of the groups. Everything in black, those are free-living organisms. Those can live on their own without being parasitic. Many of them are autotrophs, such as in the plants. And then the ones in gray, that middle ground, that's a mixture of free-living and parasitic organisms. So for the diploid organisms, these are haploid numbers. So in the case of humans, we're about 4 times 10 to the 9th. We would be right here on this scale. And you'll notice that we're sort of in the middle of the scale, and that there are many protozoans and plants that have between 10 and 100 and even 1,000 times more DNA than we have in their genomes. And then be aware that only about 1.5% of our genome is made up of mRNA genes that encode proteins. So that kind of puts a scale on things. Also, if you graph the amount of DNA in the genome versus the amount of coding DNA, that is, the things that code for proteins, 100% or pretty close to 100% agreement between the genome size and the amount of coding DNA for DNA viruses and RNA viruses as well. And even for prokaryotes, it's pretty much a 1-to-1 ratio, where about almost 100% of the genome consists of mRNA-type genes. But then by the time you work your way up into the unicellular eukaryotes, right here in purple, the number starts dropping down below 90 and 80%. And then by the time you get to animals and land plants, somewhere around 1 to 10% of the genome consists of coding DNA. And this is where you start getting more and more sequences that are small RNAs that are controlling those mRNAs, so that you're having more and more of the RNAs controlling expression in different ways, not only at the transcriptional level, but at the translational level. And much of it is repetitive DNA, including repetitive DNA from transposons, retrotransposons, and introns, as well as small RNAs that can inactivate some transposons, can control the rate of mutation, can control expression at the transcriptional and translation level. So about 45% of the human genome is made up of transposons, which includes a lot of the repeats. 84% of the maize genome is transposons. So both of these elements, including transposons and small RNAs are a diverse set, and also transposons are diverse, have been crucial to the evolution of eukaryotes, and especially the speed of evolution at times. And additionally, remember, these transposons are related in some ways to viruses and introns, and those have also been crucial to the evolution of eukaryotes. So now I want to start this move through these different genomes from different types of organisms. And the first stop will be with RNA viruses and DNA viruses. And in some cases, I'll explain quite a few molecular details about some of these. As we work our way up higher with bacteria and archaea, I won't be able to discuss in as much detail, but realize that everything that goes on down here molecularly in RNA viruses and DNA viruses also are the same mechanisms that happen in bacteria, archaea, and eukaryotes. And so you should remember all of those mechanisms as we move our way through these genomes. So let's start talking about viruses first in a general sense, and then I'll talk specifically about RNA viruses and then DNA viruses. Bacterial viruses are of 4 types, depending on the species. There's a double-stranded RNA version. This is what they carry in the virus particle. Single-stranded RNA versions, double-stranded DNA viruses, and single-stranded DNA viruses. So starting with a double strand, they already carry the positive and the negative strand with them, and they also carry with them an RDRP, which is an RNA-dependent RNA polymerase. So the positive RNA can immediately be translated into new virus particles and proteins. And the double-strand version can go through replication to form more double-stranded molecules that get packaged in the virus particles, and then they lyse the cell, usually killing it, releasing the viruses. Single-stranded RNA usually carry the positive strand, the mRNA, with them. That can immediately then form virus proteins using the translation system of the cell. They also carry along with them an RDRP, an RNA-dependent RNA polymerase that will then make the minus strand more positive strand and then produce virus particles by wrapping those up in the viral proteins and then lysing the cell. For DNA viruses, they can be double-stranded or single-stranded. They don't have to carry an RNA-dependent RNA polymerase with them, so they don't. But then you have double-stranded DNA here, for the single-stranded DNA viruses, using the host DNA polymerase to make the double-stranded version. Many of these can undergo lysogeny, not all of them, but that's integration into the host chromosome. And so then they remain quiescent for a time while the bacterium is living and reproducing. But then they can pop out by excision, using certain proteins to excise them, and then start transcribing the DNA into mRNA, new viral proteins using the translation system, the ribosomes in the bacterium, and then assembly of those into new virus particles. The either single-stranded or double-stranded uses the replication machinery of the cell to produce either single-stranded DNA chromosomes or double-strand DNA chromosomes, and then package those up into virus particles and lyse the cell, kill it, releasing all the newly formed virus particles. Eukaryotic viruses have been defined into 7 categories, according to David Baltimore. So this is the Baltimore scheme. So category 1 is a double-stranded DNA. Category 2 is single-stranded DNA. Categories 3, 4, and 5 are RNAs, both double-stranded and positive single-strand. Positive means that's the mRNA-like strand, and negative strand single-strand RNAs. And that would be the template strand for the mRNA, but not the coding strand. And then there's 6 and 7 over here. This is a single-stranded RNA virus that carries reverse transcriptase, and that's like HIV virus. And finally, double-stranded DNA viruses here that also carry a reverse transcriptase and probably are related to these over here, category 6. So categories 1 and 2, these double-stranded and single-stranded DNAs, are similar to the ones in bacteria in that you immediately build a double-stranded DNA molecule if there isn't one in the single-stranded version. They can lysogenize and remain integrated in the chromosome for quite a while. Or they can excise. They can also transcribe from the host chromosome and make some RNA, mainly to keep them in check, but sometimes they actually can form more virus particles that way. The type 3 RNA, double-stranded, has a positive and a negative strand, so it also carries along with it an RNA-dependent RNA polymerase so that it can make new strands of RNA. But the positive strand is the mRNA strand, which can be translated into viral proteins and then incorporated into viral particles. The positive and negative sense strand are RNA viruses. Use the positive strand to as an mRNA to make viral proteins. This one you see starts out right away using its RDRP to make a positive strand. Then you get RDRP making both the positive and the negative strands. The double-strand RNA viruses also use RDRP to make more double-strand RNA, and then depending on what type of virus it is, it gets incorporated into the virus particles, becoming either category 3, 4, or 5. Category 6 is a single-stranded RNA virus, but instead of using RDRP, it uses reverse transcriptase. So instead of making RNA molecules, it makes DNA molecules, so it can make a double-stranded copy of itself and integrate that into the chromosome. It can also use that for transcription to make mRNA, and then to use that with reverse transcriptase to make the other strand of RNA, which becomes incorporated into the virus particles. The mRNA is used to translate proteins, to wrap up the virus chromosome as well. For double-stranded viruses, DNA viruses that carry reverse transcriptase, they come down here. They have the double-stranded DNA that they replicate using the host enzymes. And then this is used to make a copy of the genome here, and it carries along with it the reverse transcriptase. So each one of these, let's say RT, not only package up the DNA in the virus particles, but they package up the reverse transcriptase and usually other proteins as well. So I just wanted to introduce lysogeny because we'll be talking about it later. So this lysogenic part where a copy of the viral genome becomes integrated into the host chromosome. And this occurs with DNA viruses as well as RNA viruses that carry along with them a reverse transcriptase. So in the case of DNA viruses, and it can be double or single-stranded, but immediately make it a double strand, many of these circularize within the host, and then there's a crossover event within the host chromosome, and they integrate into that point either that or there's an enzyme, an endonuclease, that cuts those sites and they incorporate into that site. For RNA viruses, there's an extra step involved, and that's to take the RNA and reverse transcribe it into a double-stranded DNA at first and then integrate into the chromosome. There are a few variations on this also that I won't discuss right now, but realize these aren't the only two possibilities. So now I want to talk about some things that are among the simplest genomes, and those are RNA virus genomes. The first virus I want to discuss is HIV, human immunodeficiency virus that causes AIDS. It's a type 6 virus in the Baltimore category system. The genome size is just over 9,100 nucleotides, single-stranded, and it's negative sense, single-stranded. And it carries along with it a reverse transcriptase. Here are 2 copies of the genome it carries, and then this little ball, that's a polyprotein. So that carries with it not only the reverse transcriptase, but it also has a protease, RNase H, and an integrase. So all of those are in a polyprotein, meaning that's a long protein, and then the protease acts to chop that up into the individual proteins once it infects the cell. Surrounding that central part are capsid proteins, and then there are matrix proteins on the outside of that, and then this red part is a piece of the host cell membrane, the human host cell membrane, and then there are proteins, glycoproteins sticking out of the membrane here. There's a transmembrane portion and a surface glycoprotein section. And that's actually made as one protein, again a polyprotein. The glycoprotein and transmembrane protein are together, and then a protease chops those into the 2 pieces, and they're assembled on that membrane. And it's those proteins, this surface glycoprotein, that interacts with a surface receptor on T cells on human cells. So here are a few micrographs, both scanning electron and transmission electron microscopy. You can see this is a T cell all covered with the viruses probably coming out or infecting that cell. Here are more of the viruses in green attacking a couple of different types of cells, and then these are transmission looking through that. So you can see that sort of funnel-shaped central capsid, and then the matrix protein around that, and then the membrane and the glycoproteins sticking out of that membrane. So here's the complete sequence of HIV-1. There are 2 versions, HIV-1 and HIV-2, and it is continued on the next page for a total of 9181 nucleotides. So that short little sequence then can kill a human. I thought that was always amazing to me. It actually degrades the immune system, and that leaves the patients susceptible to other infections and cancers, and that's usually what kills them. So here is part of the life cycle. The virus binds to CD4 receptors on T lymphocytes with its GP120 glycoprotein on the outside. And then it fuses with the lymphocyte, replicates, produces new virus particles. So there's an initial infection stage where there are kind of flu-like symptoms, mild symptoms, but then it can go on to do other things. It can integrate into the chromosome of that lymphocyte, becoming a lysogen, and it can actually then become a virus factory, or it can pop out of the chromosome and become lytic again and produce more and more and more virus particles into that patient. Eventually, it changes that cell, including the nucleus, so it becomes multinucleate, and that's called a T lymphocyte syncytium. And anytime you have multinucleate cells like that, their syncytium has absolutely no immune response, and it just becomes essentially a virus factory. So not only is it producing a lot of viruses, but it's degrading the immune system by essentially taking those T lymphocytes out of the immune system. Here is an average progression of the disease. The first 12 weeks occur like this. You get an acute infection of HIV. There's a drop in the number of CD4 T cells, but then the virus, the T cells actually start killing the virus. You get a reduction in the HIV in the blood, and there's an initial slow rise or small rise of the CD4 T lymphocytes, but eventually the virus keeps producing year after year after year. During that time, it's infecting more and more of the T cells with the CD4 receptors, and those are dropping. And over 5 to 10 years, then the CD4 levels are so low that the patients start developing other types of diseases. They're susceptible to many types of diseases then. So then after that point, there are opportunistic diseases then that eventually lead to death of the patient. Here is a slightly different view of the virus. Again, there's the capsid protein that encapsulates 2 copies of the negative sense strand of the genome, and then there are the polyproteins, which contain the RNA, or the reverse transcriptase, the RNase, integrase, and the protease. And then there's the matrix proteins that surround that here, and then embedded in the membrane are the GP41 transmembrane protein and the GP120 glycoprotein that attaches to the CD4 receptors. Here are the genomes of HIV-1 and HIV-2, slight differences between them. They have 5' and 3' long terminal repeats, which are characteristic of also retrotransposons. So these are probably related to those. And of course, those are characteristic of an organism or a genome having a reverse transcriptase. So that fits the model of a retrotransposon also. And then this first gene makes a polyprotein called GAG, which includes the matrix protein, the capsid protein, a nucleocapsid protein, and another protein, P6. And again, the protease will chop that up into the individual proteins. Second gene here, and you see they're overlapping, so it's a very condensed genome. That's the pol gene that makes the pol protein that includes the protease, the reverse transcriptase, the integrase, and an RNase as well. Then there are a few other genes, VIF, VPR, NEF. These are things that control movement of the viral RNA and the proteins during infection. And then there's this envelope gene that makes the surface protein and the transmembrane protein that sit on the surface. And it's that surface protein that attaches to the CD4 receptors. Also note here that this REV and TAT are partially encoded over here and partly there, and so there is splicing involved to make that RNA and eventually encodes for a protein. So here's the infection cycle. There's the infectious HIV virion with the surface receptors GP120 that attach to these CD4 receptors. The virus membrane, which is just like the human membrane because that's what it is, fuses with that human membrane and releases the internal capsid. Then there's release of the RNA, the reverse transcriptase, the protease, and the integrase. And then there's reverse transcription and transport into the nucleus by those VIP and VPR proteins as well. And then there's transcription out here. Initially, there's just production of new virus particles, but occasionally there's integration into the chromosome and then transcription from there, and then translation on the host ribosomes out here, and then virus assembly by using part of the membrane to concentrate those proteins, both outside and the proteins inside to surround the nucleic acids, and then the virions, infectious virions, are released from those human cells. So they can actually do a lot with only 9,000 nucleotides and about a dozen genes in gene products. So the next virus I want to talk about, RNA virus, is also a negative sense single-strand RNA virus, and that's influenza A. And it has 18 hemagglutinin gene subtypes, so there's H1 through H18, and then 11 neuraminidase gene subtypes, N1 to N11. So often you'll hear these referred to in the news, H1N1 and things like that. Here are the organisms that influenza A infects, all those strains. All the strains infect one bird species or many bird species. So that's H1 to H18 and N1 to N11. So birds, and especially waterfowl, are the major vectors for these viral strains. Humans tend to get either H1N1, H2N2, or H3N2, and a few other of the H1, H2, and H3 types. They can also contract H5, H9, and H7 from birds mainly. It's usually ducks or something like that. And it will infect humans, but then you cannot get, or usually don't get, human-to-human transmission after that. So it's just localized. However, if you contract those, they have a pretty high death rate. And as you can see here, chickens get many of the same strains that humans and other birds get. Cats can get H5N1 mainly, marine mammals, terrestrial mammals, and especially there's this connection between ducks and chickens and things like that, domestic, and waterfowl visiting those, and then swine as well as humans. So you can get this movement of these viruses and create new types, new subtypes, by mixing them in usually a pig or a bird. So based on their sequences, here is a phylogram and a grouping into group 1 and group 2, and the things that have the circles, H1, H2, and H3, those are the primary ones that infect humans. Usually H1 and H2 are more severe, and H3 is less severe, but all three can be severe in some cases. And then the triangles are primarily the avian subtypes that affect humans, and that's H5, H7, and H9. All of those can have pretty high mortality rates. So this is pretty much showing that there's a high degree of genetic variability among the influenza A strains. And even among things like H1N1, there's quite a bit of genetic variation just in that group as well. These change rapidly, and it's difficult to come up with a vaccine every year when they're changing so rapidly and there's so much genetic diversity. Here are a couple of views of the virus. This is a transmission electron microscope picture of the viruses. You can see they're kind of odd-shaped because they're enveloped by the membrane that they picked up from the host. There are 8 chromosomes here, 8 single-stranded negative sense RNAs, and then there's an RDRP that they take with them, a RNA-dependent RNA polymerase, the matrix protein, phospholipid bilayer, and then the hemagglutinin trimer, and the neuraminidase tetramer, and then there's also an M2 ion channel that's important for infection. But it's primarily the hemagglutinin protein that attaches to a receptor on the host cell. Here are the 8 chromosomes. So on chromosome number 1 up here, it's the PB2 protein, which is a transcriptase. and RDRP. Here's another part of the transcriptase enzyme, PB1, as well as PB1F2, which inhibits mitochondria, so it causes cell death. And then this is the transcriptase and protease is the next chromosome down. This is encodes the hemagglutinin, this one HA, a nucleoprotein that binds to the RNA. And then the neuraminidase protein here, the neuraminidase gene and protein. And finally, two shorter chromosomes. They contain the M1 and M2 genes for those proteins, which are structural proteins and integral membrane proteins, and then that ion channel, and I'll show you in a few minutes what that does. And then a couple of non-structural proteins involved in RNA transport, splicing, and translation, as well as an export protein for exporting the viral RNA from the nucleus. So here's the infection cycle. You have the infectious influenza A virion. It attaches by the hemagglutinin protein on the outside to sialic acid residues on the outside of the host cell membrane. And then you get endocytosis of that virion. And then the ion channel, the M2 ion channels, acidify that vesicle, and it breaks open. And in breaking it open, then the viral RNA and the RNA-dependent RNA polymerase start replicating that RNA. They make the positive strand as well as the negative strand in the nucleus. Those are transported out, and you get translation of the positive strand out here into the viral proteins. The negative strands are incorporated, one of each of the 8, into that particle, and then there's budding off of the infectious virus particles from there. And you'll note over here that the sialic acid has been clipped off of those receptors. And so what happens is there's a protein that is produced by the virus RNA that clips off the sialic acid residues before they can be exported out of that cell. So that means that then as these things bud off, they don't stick again to the same cell, but they can only infect a cell that's not been recently infected so that they still have their sialic acid residues. So what makes influenza A so variable, have so much genetic diversity, is that you can get recombination on each one of those chromosomes to create new versions. But you can also get something called reassortance, caused by a reassortment of the 8 chromosomes. So this is something that happened in 2009 with the swine flu. There was a pretty bad one. I got that one, and it was a very bad flu. And they've traced back what was in the 2009 H1N1 human H1N1 flu, the swine flu. It contained some fragments, some chromosomes from swine, but it also had some chromosomes of human origin, in red here, and from avian origin, in purple here. So it was a reassortant. And those sometimes are the most dangerous ones because they pop up out of the blue, and no vaccine has yet been made for them. So the scientists have to then start finding a vaccine for those specific ones. It looks as though a few years before that, in 2000, there were also a couple of reassortant varieties that came together to form that 2009 version. And if you go back further, they were a combination of a Eurasian swine flu here with a fairly pure swine flu back here, H1N1, back in 1990, a fairly pure H3N2 from humans, and a fairly pure avian strain in 1990 that then mixed by 2000 and then reassorted again in 2009 to become that 2009 swine flu, which was quite deadly. It had a fairly high mortality rate. And again, there are 8 chromosomes, and the total of those is 13,600 nucleotides. So the next RNA virus then is Ebola. Again, it's a negative sense, single-stranded RNA virus. Some of the worst ones are. And it infects by first creating the mRNA by using an RDRP, RNA-dependent RNA polymerase. It also makes positive strands and then packages makes a negative strand from there, packages the negative strand into the virus particles by budding off of the cells, and the proteins are produced by translation using the host ribosomes. The disease itself causes the cells to rupture pretty much, and so that's why there's a buildup of fluid, because all of that cellular fluid is leaking into the surrounding tissues, and so people tend to bleed from many places on their bodies. And here's the structure of the virus and the genome. The genome is about 19,000 nucleotides on one piece of negative sense RNA, single-stranded. So here's Ebola, and here's a related virus called the Marburg virus, causing very similar symptoms, and they seem to have been around both in Africa and Europe for many hundreds of years, so it's not just a recent arrival. Here are the peplimers, which attach to the cell surface of the host cell, the human cell, and then the envelope is the membrane that it gathered from the host cell. Matrix protein, a nucleocapsid, and then a long linear RNA molecule. So there's the nucleocapsid protein, a glycoprotein, which is the peplimer. These VP35, VP40 are proteins that are used for movement of the RNA in the cell. And actually, this one never goes into the nucleus. It stays out in the cytosol. And there's the large or long gene, which encodes for the RNA-dependent RNA polymerase. So the last RNA virus I want to talk about is the coronavirus. As you know, the one that's going around right now is the SARS-CoV-2, which causes the disease COVID-19, 19 for 2019 when it was first identified. So the coronavirus looks like this. There's one long RNA. It's a very long RNA, almost 30,000 nucleotides. So this is one of the largest or longest RNA genomes among viruses. And then there's a membrane around here from the host, and then there are different membrane proteins, including the spike glycoprotein, and those are the ones that attach to the receptors on the host cells. Here's a view of them in the electron microscope. You can see those spike proteins sticking out, and when these were first found in the 1950s or 60s, I believe, they thought they were influenza viruses, but they turned out to be something quite different, actually. So here's the infection cycle of both SARS, which is the sudden acute respiratory syndrome virus, and MERS, which is the Middle East respiratory syndrome virus. Both infect the same way. This one attaches to the ACE2 receptor, and this one to a different receptor, so that makes the infection different. This one will attach to human cells as well as bats and things like this. And this one actually will attach to camel cells as well as human cells. So a different type of attachment to different species. So they're enveloped by the host cell, and then they start coding. Then they release their viral RNA into the cell. You start getting translation right away. Makes the replicase, which is an RDRP again, and then eventually you make also the negative sense RNA, which is the genomic type, which gets put together with these proteins that are made down here, including the spike protein, which is that S, and other proteins that are translated on the rough ER, in the rough ER, because they wind up stuck into a membrane. And so those eventually form a capsid around, and then the envelope around that with the spike proteins and other ones in there. And then that's released from the cell from there. MERS actually has a very similar pathway, and it also has a spike protein, but it has a different epitope, that is the protein part that's at the surface that's being attached to the receptor, and that's why it attaches to a different receptor. So this is CoV1, and now we have CoV2, which is the one that's currently circulating and causing this pandemic. And its spike protein is also a little different in that it's able to hide, essentially for a while anyway, from the human immune system. So here are the genomes of both SARS-CoV, and SARS-CoV-2 looks very similar to SARS-CoV, and MERS-CoV down here. And you can see this one's about 30,000 nucleotides, and SARS-CoV is approximately 29.7 thousand nucleotides. So there are open reading frames here. There's the spike protein, and then there are all these other proteins, some of which become embedded in the membrane, including the E protein and the M protein and the N protein. And it's this spike protein that exhibits the most change, the most variability, and that's the one that attaches to the receptors, and that's the one that makes things kind of dangerous as far as moving from one host, like bats, into another host, such as humans. Here's another view of SARS-CoV and MERS-CoV with a few diagrams here showing some of where those proteins wind up. So these open reading frames up here, ORF1 and ORF2, are primarily for proteolysis, that is cutting a polyprotein, viral replication, that's the RDRP, that is RNA-dependent RNA polymerase, and viral replication, a helicase there. And then all of these down here are those surface proteins, including the spike protein, E protein, M, and N proteins, and those all wind up in the membrane. And some of those are ones that sort of exacerbate some of the immune system to kind of go wild, and that's usually what causes the most damage, in that there's what's called a cytokine storm, where the immune system, parts of the immune system, overreact. And that happened in the 1918 influenza also. People sort of drowned because their lungs were filling up with fluid, mainly a response of the immune system caused that to happen. Initially, some people proposed that this SARS-CoV-2 was a human construct, and that it had been made for a bioweapon, essentially. But this study came out and showed that that's very unlikely. So they compared the SARS-CoV with some other coronavirus, including some from bats, pangolin, another human, the SARS-CoV, CoV-2, and some other bat sequences. And each one of these little blocks shows where they're in common. The other ones are a little more different. So this one, that set of amino acids there, groups together for the human, bat, and pangolin. And then these ones group together with the human pangolin and a different bat SARS, and then again bat or human and pangolin. So it's clear that that this moved probably through pangolins, through bats, and then through humans. And it may have been from someone eating a pangolin or something like that that was infected, that may have been, case number one. Another important part of this figure is this, and that shows an insert that's only found in human SARS-CoV-2. And this part of is part of the spike protein, sort of curls over in a way that was unpredictable. Like a scientist wouldn't predict that piece would go in there. And that part causes the spike protein to somewhat evade the immune system for some time. So that indicates that this thing had been evolving, changing inside of humans for quite a while before it really popped up and became noticed as a disease. So those characteristics, that is, it's related to a bat and pangolin, and more so to a pangolin a virus than to a bat virus, but also the bats are probably involved as well. And the fact that it had probably evolved and changed inside of humans, been selected for because of that insert, and it could hide out in the humans, hide out from the immune system, means that it most likely was not a lab-produced strain, but it was a natural strain and a product of just evolution and selection between the animals and the humans. So if you look historically, there are a lot of these SARS type viruses that have popped up in China. This one in 2003 was an unusual respiratory disease, identified later as a SARS, sudden acute respiratory syndrome is what it was called. And a very similar virus was found in some caves, bats in some caves near that same site. And it's where some Chinese would collect wild animals from that region to sell in live markets. So shift about a decade and a half and a couple of 100 miles, and now there's a new unusual respiratory disease that looks like it popped up prior to the middle of 2019, and maybe even before that, maybe in 2018, but no one really noticed. And this is the start of the CoV-2. And just like the 2003 SARS virus, it was within the vicinity, not, a couple 100 miles at least, from caves where bats were known to carry these coronaviruses. But also pangolins were native to that region as well. But by late 2019, it was recognized that this was a respiratory disease spread through the air, and that it was a brand new virus with no human immunity. So it started as an epidemic, and very, very quickly, because of all the airplanes flying all over the world, it became a pandemic. So it moved from China into Europe, into other parts of Asia, eventually into Africa, into the US, and into South America as well. And when I first put this together, so this was somewhere around summer of 2020, already 8 strains of CoV-2, so it had already changed quite a bit. 8 strains were being tracked, and I think there are up to 11 or so now. And some of them were more virulent than others. So the one from Europe, which came over to the U.S. also, especially the eastern U.S., was more virulent than the one in China that moved to the West Coast. So there were differences in the sequences as well as their effects on humans. Okay, so that's all I want to talk about RNA viruses, some of the smallest ones, and I talked about some of the largest ones up here. And next I want to talk about DNA viruses. So as with the RNA viruses, I will start with the smallest DNA virus genomes and move up to the largest DNA genomes. Here is the first DNA virus. It's a single-strand DNA virus, Phi-X174, and has about a genome size of about 5,360 something nucleotides. So here's the infection cycle where it infects a bacterium by attaching the H protein to the surface of the bacterial outer membrane. And then it injects a single-strand DNA into the bacterium. It circularizes and then produces a double-stranded RF, or replicated form DNA. Then you get transcription off of that to start forming the viral proteins, which form the major part of the virus particle. So the F, G, B, and D proteins come together to make this initial virus particle. And in the meantime, the A and A star proteins, which are polymerases, begin a rolling circle type replication by reading off of one strand and then injecting or loading up the head here with the minus strand of the DNA. Finally, it finishes up, and often slightly more than one genome's worth is put into the virus particle. Finally sealed up, and then the H protein is attached to the outside, and finally this E protein lyses the cell, and then you start the infection cycle again. So as I said, it has a small genome, 5,368 nucleotides, and 11 genes if you count these two separately. But you can also see on these viruses the genes are very much packed together. In fact, they're overlapping. So you have a high concentration of genes. Over 100% of the genome is coated with genes, and that's if you say that these are double genes essentially. So here's the origin of replication here, where you start getting replication of the DNA. This is the A promoter, which then starts transcribing and then eventually translating the A and A star polymerases that replicate the DNA of this virus. Then there's another promoter here that begins transcription of the B and K genes. And then there's read-through all the way through to make C and D. So you'll note here that the A gene and A star genes are transcribed first, then those are translated into the proteins, and after that you get C and J as well as B and D. And so you're starting to form the capsid and the DNA in those early stages of transcription and translation. And so then finally the F, G, and H are produced. So this is completing the capsid of the virion. And then this E gene and E protein finally control the lysis of the cell to release the viruses. So there's a coordinated expression of these genes. Also note that these are terminators of transcription, although there is a slow read-through of those transcription stop sites. So although a lot of them stop here, some of them go through here, some of them stop there, they go through a little further, stop there, and go further. So there's a sort of a mechanism for producing the proteins to build the virus structure, proteins to replicate the DNA and fill the head, and then proteins to lyse the cell and control when lysis is done. All of that within those 11 genes and 11 protein products from the genes. The next genome I want to talk about is bacteriophage lambda, which is the genome is about 10 times larger than that of Phi-X174. So here's the long linear chromosome of 48.5 kilobases, and it has about 56 genes rather than just 11 for Phi-X174. And this is a double-strand DNA virus rather than a single strand. So on this end, the left arm, you have mainly genes that will produce proteins to produce the head and tail of the virus. In the middle here, you have control regions, which contain 9 genes, 4 promoters, 2 operators, 4 terminators, and the origin of replication. And then a couple genes here for integration to produce the lysogen of the virus, and one here for mainly for lysis. Once the DNA, here's the DNA replication. Once the DNA is produced in the heads and tails, then you've produced a virus, and then lysis of the bacterial cell. There's a region in the middle of unknown function, and actually if you delete that, it has no effect on the virus. So it appears that less than 100% of the virus genome contains genes, although, in fact, that's used a lot for cloning. So people will insert pieces of DNA of interest into there and then clone those by infecting with the lambda phage into bacteria. The virus injects its DNA into the bacterium as a linear molecule, but shortly thereafter it circularizes around these COS sites, which are cohesive ends. So there's a COS L for left and a COS R for right. So there are the head and tail genes that wind up here on the circle. And then here's this unknown region after the tail genes up here. Here's the integration and excise functions here, and then recombination genes here, and then these control regions that contain C1, C2, C3, N, and CRO. And these have a lot to do with which direction the bacteriophage goes. Does it go towards the lytic cycle, making new heads and tails and DNA to then lyse that cell, or does it become lysogenic and go in this direction and integrate into the chromosome? So this control region controls that function, whether it goes one direction or not. So this is a virus, a bacteriophage of about 49 kilobase pairs, as it's double-stranded. But you can see there's already quite a bit of control, a complex control of expression of those genes. So remember this sort of complexity. I'll go through it in a little bit more detail, but remember this in the virus. And these same sorts of things are happening in bacteria and eukaryotes, but on a larger scale. So I'll go through the details here, but realize for those other organisms with much larger genomes, I won't be able to provide as much detail. As I mentioned earlier, bacteriophage lambda can go through a lysogenic cycle. And so when it first enters the cell, it will circularize around those COS sites. And then there's an at site. At lambda will attach to at B. So these are regions that are somewhat homologous, so you can get those lining up and getting a crossover in there. And the lambda prophage then is formed by integration of the lambda genome into the E. coli genome. And it will remain in that lysogenic state for sometimes quite a while, sometimes almost forever for that bacterium. And it can later excise out by turning the excision genes on. So it's a different set of genes over by the integration, the int genes that integrate that into the chromosome. So that's the lysogenic cycle. Then at some stage it can excise here and enter a lytic cycle. And so it turns on the early genes to go the opposite way around the chromosome to then first make a theta form type of replication to make a few copies of the genome, and then it goes through a rolling circle type replication after that. And in the lytic cycle then there are those COS sites, so you get a long continuous concatemer of the genome of the chromosomes. And then there's actually a protein A which sits there on that COS site. And once it gets to a full head's worth and it finds the COS site, it will cut at that site, and the head will be then completed, attached to the tail, and then many more of those are produced in the bacterium, and then the lysozyme will be produced, and it will lyse the cell, releasing all of the newly formed phage. So when those phage then infect another cell there's actually a decision that's made at that point. Does the phage, the virus, go through a lytic cycle next, or does it go through the lysogenic cycle next? So remember that control region on this chromosome, that's where that decision is made, and it's based partly on the health, the nutritional health of the bacterium, and the temperature, whether the bacterium is under stress. If the bacterium is under stress, usually it'll go through the lytic cycle so that it can try to produce more virus particles and infect bacteria that are not being stressed at the time. Here's a close-up of that control region. So out to the right here, those would be the early genes to go towards the lytic cycle, and to the left over here, the genes over here would be int and excise and recombination genes and things like that for the lysogenic cycle. So just like we saw in the lac operon in bacteria, there are promoters and there are repressors that control the expression of those genes and sometimes many genes at the same time. So if we start at this promoter here, PR and PM, one goes in one direction and one goes in the other direction, and they're controlled by C1 protein and CRO proteins. So if C1 is produced in higher amounts, then it actually sits on the promoter and will not allow CRO to be produced and to go in that direction. On the other hand, if more CRO is produced, it will sit on that site, not allowing leftward transcription over here towards the lysogenic cycle. So as I've said down here, if the CRO protein is produced in higher amounts than C1, then transcription proceeds through Q, and Q turns on the PR prime promoter, and you go towards the lytic cycle. If CRO is less than C1, that is, C1 is in higher concentration, then lysogeny is established because you go in this direction. If C1 breaks down eventually, so it is even in the lysogen, it's produced in small amounts to control transcription. But if C1 breaks down, then excise and some other proteins that interact with the DNA to chop out that lambda DNA. Then the lytic cycle is initiated. So then you start here and go in this direction, and that's to end lysogeny. So that's that decision area. There's a decision to be made to go either lytic or lysogenic, and even when it's a lysogen, there's a decision whether to switch towards the lytic cycle and come out of lysogeny. So even in this fairly simple virus, and in bacteria as well, there are these regions that are pretty highly sophisticated molecularly that control transcription of genes to go in one direction or the other, or to turn on some genes and not others. And so as we work our way to more complex organisms, remember this particular example. So here it is with a few more details added. So here's that control region. There's the C1 gene, there's the CRO gene, and this is the promoter here, the PR and PRM promoters, which go in one direction for PR, and PRM goes in the opposite direction. And C1 will inhibit transcription in the rightward direction, and CRO will inhibit in the leftward direction. I want to also point out that there's two things here, R minus and R minus. Those actually are RNAs that are controlling RNAs also. So this one, and this R minus, is one that will stop transcription of this gene Q in that direction. So again, remember that even in viruses and viral genomes, it's not only proteins that are interacting to control gene expression, but there are some RNA molecules, some small RNA molecules that are also acting to control expression. So here's that site here. There's the PRM going in the leftward direction and the PR promoter rightward going in that direction. And CRO has an attachment site to the PRM, so it inhibits transcription in that direction. It also sticks a little bit there and a little bit there, so there's 3 operator regions where these proteins stick. C1, on the other hand, attaches as a dimer and attaches mainly to this operator. A little less to that one and almost not at all to the third one over. So in the different scenarios, you start here with PR and PRM. You get transcription of CRO and C1, and then this goes into C2 and then the N. But at this point, if there's more CRO produced, then it will stop that leftward transcription that goes to the lysogenic cycle. Instead, the rightward transcription will continue and take the virus into the lytic cycle, eventually killing the bacterium. However, if C1 is in higher concentration, then rightward transcription will be stopped and leftward transcription will continue producing the lysogenic cycle and integration of the lambda genome into the E. coli bacterial chromosome. Here is a diagram of the lytic cycle of lambda. So you start with several gene products from gene B, C, E, and nu3. And then there's a GRO-E, which is a host protein that's used in this process to make a prehead. Then A, which threads the DNA into the head, and nu1, which participates in that, starts threading a genome's worth of the chromosome through, and A also recognizes the COS sites, which is where it cuts, so that there is just a genome's worth of the DNA in the head, because only a certain amount of DNA will fit in the head. However, if there's too little DNA, the head collapses, actually. Then D here puts on a few units on the bottom here that gets it ready for the tail. F2 then plugs the tail region, and then W attaches the protein W attaches the head to the tail. To make a tail, you start with the J protein, which is the lower portion of the tail, and then I, K, L, G, H, and M. There's quite a few here, which seems like, oh, it's only adding a little bit, but some of the proteins down here make that an attachment site to the bacterial membrane, the outer membrane. So it's actually a very important region for recognizing when the tail is attaching to an E. coli cell rather than any other bacterial cell. And then the VU proteins build up the rest of the tail. The Z finishes that off by attaching a piece here in conjunction with the W protein to make a complete head. And then the lysis gene turns on to cause a breakdown of the cell wall and lysis of the bacterial cell. So the last DNA virus I want to talk to sort of crosses the line between bacteria and viruses, and that's why I've chosen this one. So it's a mimivirus in the mama virus family, and then it has a Sputnik satellite virus too. The mimivirus genome is about just over 1.2 million base pairs, so it's a very large genome, which places it, as far as size, in the range of some bacterial genomes too, but only usually parasitic bacteria, ones that are obligate parasites. So here's the virus itself. It has the capsid, these fibrils sticking out, and then inside there are two copies of the genome, the DNA inside of that, and then there are core proteins and capsid proteins outside of that. It also has a satellite virus called Sputnik, and it's a tiny little thing, a much smaller genome. When the Sputnik virus infects along with the Mimi virus, then when you get packaging, the chromosome of the Mimi virus is not formed and is not encapsulated, and a virus particle is not built around that. Instead, you get several of these Sputnik viruses that are enclosed in the capsid and the fibrils. And so when both of them infect at the same time, almost all of the particles coming out of them contain the Sputnik virus only. When Mimi virus infects alone, then you get only Mimi virus and no Sputnik at all. By the way, many viruses have satellites viruses also. So the satellites are smaller usually, I think always, and they can only infect if the main virus also infects. And often they are produced in higher numbers than the main virus is. The main virus is if it infects alone, then it's only that primary virus that is produced. But when the satellites are produced, I know in the case of tobacco necrosis virus, then 70% of the particles that come out are the satellite viruses, and 30% are the main T and V virus. And that's a plant virus. So the proper name of this one is actually the Acanthamoeba polyphaga Mimi virus. And as you can tell by the name, it infects amoebae and only affects amoebae. And amoebae are a sister group to fungi and animals, so they're not too far away from us. So the first thing you do is here, the Mimi virus is phagocytized, and it sets up its own vesicle inside of the cell, which looks sort of like an invading bacterium rather than an invading virus. The DNA is transferred into the nucleus where it is copied, and then the virus DNA and RNA are transferred out to the vesicle. And then that vesicle becomes a factory for virus production, and eventually the cell is lysed, releasing the viruses. So here are a few electron micrographs of that. There's the vesicle being set up. This is the nucleus over here. And there are the virus particles being produced. Here's one virus particle showing where the chromosome is in the center and all these fibrils coming off it. This is another view of it just starting the infection phase and in a different cell. And this just shows the comparison. There's a Mimi virus and there's a Mycoplasma bacterium. When these were first seen in the amoebae and first identified, scientists believed that they were bacteria, but now we know they're not bacteria. They're actually viruses, although they have a lot of characteristics of bacteria. So here are a couple of other micrographs of this. This is that vesicle being set up, and then you can see the viruses being assembled. They're viral pieces being formed here and then shipped out here to build those viruses. And here on the lower right here, that's a full virus being formed with the DNA inside the 1.2 megabases of DNA. Now, when it infects with a satellite virus, you get something that looks like this instead of this. And you can see those small particles inside. Those are the Sputnik satellite viruses that are packaged into that virus particle rather than the normal genomic DNA from the virus itself. So this is a diagram of the infection of the Mimi virus. So the Mimi virus can infect alone, and in that case its DNA is sent here and it forms the Mimi virus particles inside that vesicle that it builds. If the Sputnik virus satellite virus infects with the Mimi virus, then the DNA is built up, but primarily the Sputnik DNA is produced. Then these vesicles are set up, you produce Sputnik, which are then incorporated into the virus particle, and the main Mimi virus chromosome is not incorporated into those virus particles. So you get these particles containing Sputnik virus rather than the Mimi virus genome. So I won't show you a picture of the chromosome of the Mimi virus because it's just too large and you can't see any detail about which genes are where. So in this case, I've made a pie chart and categorized the genes that are in the Mimi virus genome. And I'll do this with bacteria also, because the genomes are just too large to show, except in one case. So a large proportion is set up for replication of the DNA, and you might expect that, although that's a fairly large section of the genome for replication. Now, this differs from most viruses in that now you have genes for transcription and translation. Usually host systems are used for that. And then there are genes for general cellular function, which also is not common for at least small viruses. And then post-translational modification, unknown. Some of these are the proteins for the capsid, etc., and other functions for the capsid. Then nucleotide transport and metabolism. So that's unusual for a virus also. A few have some of those, but not all of them. But then one set that's really bizarre is cell wall and membrane synthesis. So usually a virus isn't going to build a cell wall, a bacterial cell wall. The bacteria build those. Then you have signal transduction proteins, RNA processing, secondary metabolites, amino acid transport and metabolism, cell cycle control, etc. Those are things that you expect to have in a cellular organism, but not in a virus. So what this suggests is that the Mimi viruses may have evolved from a parasitic bacterium, and their genomes are still undergoing reduction. So that sets the path for a lot of other viruses too, and that's been a hypothesis for a long time, that viruses are just reduced parasitic organisms, and some have reduced only to a few genes. In this case, this virus has hundreds of genes, a 1.2 million base pair genome, and has a lot of genes for cellular function. And so it may be that it's sort of a virus on its way towards evolving a smaller and smaller genome consisting of fewer and fewer genes, just to get down to the essentials that still build the virus, but don't do much else. On the other hand, the satellite virus is very much reduced. So it's about the size of a Ebola virus. But again, this is a double-stranded DNA virus. Here's the Sputnik virus, consisting of 21 open reading frames or genes. And several of those have been identified. So this number 10 is an integrase that would integrate that sequence into another chromosome. There's the major capsid protein. Also, there are genes for primase and helicase for replication, ATPase, a zinc ribbon, a DNA binding protein, and an insertion sequence, a transposon. It also had been noticed that there are these double-stranded loops possible, usually in between genes. So they may be there for control of transcription or expression of some of those genes. Okay, so far in our trek through genomes, we started down here with very small RNA viruses up to one of the largest RNA viruses, the coronaviruses. and then talked about some small viruses all the way up to the Mimi viruses up here. In the next set of videos, I'll go through bacteria and archaea, and then in a subsequent video, I'll talk about parasites, endosymbionts, and organelles, and then finally into eukaryotes. Okay, so that's it for this initial video on genomes and genome organization and evolution. So I'll see you in the next video.